Now let's look at a dihybrid cross in detail. So once again, Mendel here is looking at two different traits in the same organism. In this case, yellow peas that are round versus green peas that are wrinkly. So green is recessive and wrinkly is recessive. So we can see that. Now, here's the question. Well, here's what he sees. He breeds these two together, and he gets peas that are yellow and round, right? Big Y, big R, little r. Those would all be yellow and round. Then he breeds those with themselves or with each other to get the F2 generation. And he comes up with two predictions, right? So he has a hypothesis, or a couple of hypotheses, of two hypotheses as we see here. One is dependent assortment. The other is independent assortment. Those are the hypotheses, and then he has his predictions, what he would see if those hypotheses are correct. Now, dependent assortment means that dominant traits are inherited together. So big Y and big R would always be inherited together, and little y and little r would be inherited together. So in that case, this organism could only make big Y, big R sperm and little y, little r sperm. And if that were the case, once again, you might end up here you can see big Y, big R, little y, little r, big Y, big R, big Y, little r. Here are the offspring. And if that were the case, you could get a phenotypic ratio of 3:1. 3 yellow rounds to 1 wrinkly green, if it was dependent assortment. The other hypothesis was independent assortment. And if independent assortment is true, then sperm or egg could have, from this individual here, could be big Y, big R. It could be big Y, little r. It could be little y, big R, or it could be little y, little r. So there are 4 gamete possibilities if the hypothesis of independent assortment is correct, leading to this prediction of offspring. And so you can see all the possible offspring here. If independent assortment is true, he predicted a 9:3 to 3:1 offspring ratio, meaning 9 round yellow to 3 round green to 3 wrinkly yellow to 1 wrinkly green. And so he does the experiment, and he gets 315 of these to 108 of these, right, to 101 of these to 32 wrinkly green, and that comes out as pretty much a 9:3 to 3:1 ratio. So this is the model that holds true. Independent assortment. So that means that these traits are inherited separately. Probably they're on separate chromosomes, right? And I would like you to try this yourself, and I'll get you started. Here is a dihybrid cross. We have tall purple plants that are breeding here. They are both heterozygous for both traits, heterozygous for tall and purple. So first of all, what gametes could they produce? Can you come up with that? What gametes would these be able to produce? Well, they can make a big T and a big P, right? They can make a big T and a little p. They can make a little t and a big P, and they can make a little t and a little p, right? So what you do then is you put those gametes down here. Big T, big P, big T, little p, little t, big P, little t, little p. And then over here you would have the same big T, big P, big T, little p, little t, big P, and little t, little p. And then you start crossing them together. So for example, this first box would be big T, big T, big P, big P. If I do this one here, it would be big T, big P, big P, little p. All right, so you can fill out this entire chart yourself, and then I want you to count and actually put down how many of each of these phenotypes you find. That is your mission. But that is a dihybrid cross. You can even do trihybrid crosses, which I will not make you do in this semester. But remember, if this, let me go back. If this were a, if instead of independent assortment, meaning if If dominant traits could only be inherited together and recessive only together, then the only two gametes possible here would have been big T, big P, and little t, little p. But Mendel found that that was not the case. And we now know that is not the case because alleles are these genes are actually found on separate chromosomes, so they're not inherited together. Now, I want to show you an example of a trait that is inherited in a Mendelian way in humans, and that is sickle cell. So in humans, we have 2 chromosomes that contain the gene for hemoglobin, right? And there is a sickle cell type and a normal type. And if you have 2 copies of the sickle cell type, then all of your blood cells have hemoglobin that's a different shape than normal. And those hemoglobin cells, because they're abnormal, they interlock together like puzzle pieces, and they're not supposed to do that. And then that causes the whole red blood cell to change shape, and that leads to sickle cell disease. Individuals with cardiovascular problems at a young age and things like that. A lot of pain and short lifespans. These are emergent properties from simply a change in DNA to the full-blown disease. These are emergent properties as we move up the biological hierarchy. Now, in individuals that are heterozygous, they have a sickle cell allele and a normal allele. Some of their their proteins are actually sickle cell hemoglobin, and some of them are normal. So in this case, rather than being completely dominant or recessive, you actually get both expressed, and you end up with both kinds of cells possible. But what can occur is more cells will go kind of sickle shaped in the body when malaria is present, and that allows the body to fight malaria more readily than if you don't have sickle cell or you're not a carrier for sickle cell. And let me add in a little more terminology here. If you are a carrier of a trait, like let's say you're big T, little t, so you have the little t trait, that means you're a carrier, right? That means you're a carrier of that trait. This person is a carrier for sickle cell. They could have children with full-blown sickle cell, but they are a carrier for sickle cell. This is how a trait could skip a generation, right? So this person doesn't have it, but they have an offspring that does have it, right? So a lot of traits are this way, but some of them are more complicated than the way Mendel studied. We have a lot of traits that are more complex than dominant recessive in humans. Sickle cell is one of them, because even if you are a carrier for it, some of your hemoglobin still is sickle shaped, or not sickle shaped, but some of that hemoglobin is still sickle cell hemoglobin and can cause sickle shaped cells. But that's actually potentially a good thing. Here we can see where sickle cell is really common. Here, here, and here, right? And that's also where malaria is really common. And sickle cell, when you're a carrier for sickle cell, it helps you actually fight malaria. Whereas if you don't have a sickle cell gene, you are far more susceptible to malaria. So here we can see that it's a co-dominant allele. It's called co-dominant rather than recessive dominant because they both show up. So if you are, A is normal hemoglobin, big A, right? But if you are a carrier for sickle cell, that's big A, little a, and you have offspring with someone who's a carrier, big A, little a, there's a 1 in 4 chance that your child will have full-blown sickle cell anemia disease. There's a 1 in 4 chance that your child will not be a carrier for sickle cell, and there's a 50% chance that they will be a carrier for sickle cell. And 1 in 10 African Americans is a carrier of sickle cell. So something to keep in mind. Other traits that are inherited in a Mendelian type fashion are color blindness. So if you look at this screen and you don't see a letter 8 in the middle, that could be a sign that you have color blindness. Don't use my one slide as a definitive factor there. Go see an eye doctor. But color blindness, interestingly enough, is inherited on the X chromosome. It's an X-linked trait. You might hear that term, X-linked. And show that here with this letter C next to the X chromosome. So if you are, what I want to show you is we have a dad who is not colorblind and a mom who is a carrier for colorblindness. You have to have both copies if you're a woman to be colorblind. And you can see that in this case, they will have one daughter who's a carrier for colorblindness, and they will have one son who is XCY. Now this son is colorblind. Why? Because he only has one X chromosome. So if you just have one colorblind gene as a male, you're colorblind. And I bet a lot of you know of colorblind men or are a colorblind man, but I bet many fewer of you know of a colorblind woman. It's less common because to be colorblind in a female, you have to have this genotype. So that means dad would have to have been colorblind and mom would have at least had to have been a carrier for colorblindness if you were going to have a shot at producing a colorblind female. So practice these. Draw this out. Make sense of it. Don't just stare at this lecture slide and hope it makes sense, right? But what's cool is we can take what we've learned from Mendel and we can then map it on top of what we know about meiosis. We can say, okay, so yellow round seeds, green wrinkle seeds. So there's all these chromosomes here. Some of them have the yellow, the color gene. Some of them have the wrinkly gene. And then when they make gametes, we end up with gametes that only have one of each in them, a color gene and a wrinkly gene. They fertilize the next generation. We end up with the F1 generation, which undergoes meiosis, producing all of these potential gametes. Big Y, big R, little r, big Y, little y, big R. And when those come together, we can end up with that 9:3 to 3:1 ratio. And this figure also shows you the law of segregation, the law of independent assortment. I really recommend you look at this and make sense of it, okay? If you can put this all together, it will really help you out. And this figure is in the next chapter, the very beginning of the next chapter of your textbook. So to see this figure, go ahead and flip forward to page 241, or if you're on the e-text, it's figure 12.2 in your textbook, okay? Check that out. So I'm going to post some Mendelian genetics questions. You have a lab on Mendelian genetics as well. I want you to work with this information, work on the terminology, and practice it. All right. I believe that's it for today.